What actually is muscle hypertrophy? In order to view hypertrophy from a zoomed out perspective, it's helpful to first zoom in and understand the process from a cell bio point of view. As you likely already know, your body is made up of a bunch of cells, each of which is composed of organelles that serve specific functions. Today we're going to zero in on an organelle called the nucleus, which serves the function of housing your DNA. You can think of your DNA as the master recipe book for building proteins, which you can think of as the workers of your body that facilitate all of the building, breaking down, converting, and transporting of materials that occurs in and outside of your cells. The journey to building a protein, referred to as protein synthesis, starts with a process called transcription, in which a group of enzymes makes a copy of a specific portion of DNA that serves as a specific recipe for a specific protein. This copy is called RNA, specifically mRNA. Next, that copy is shipped out to a ribosome, another type of organelle, which uses the instructions from the recipe to link a bunch of amino acids in a particular order into a chain called a polypeptide. This portion of protein synthesis, where the polypeptide is formed, is known as translation. Once the polypeptide is built, the chain undergoes a process known as protein folding, where it is folded onto itself and then again onto a bunch of other folded polypeptides to form a protein as the ultimate structure. So what does this have to do with making gains? Well, if we took a muscle, say a bicep, and chopped it in half, we would find a bunch of bundles called fascicles inside. And inside those bundles, we would find a bunch of myofibers or muscle fibers, which are the actual muscle cells of that bicep. Then if we continue this dissection, we'll see that each muscle fiber is a conglomeration of myofibrils made up of myofilaments, which are proteins that serve as the actual contractile units of the muscle. In other words, a muscle's motion is the sum of the motion of the millions of myofilaments composing the muscle's underlying structure. Now, there are many proteins involved in this system, such as myosin, actin, and titin, and they're arranged into functional units called sarcomeres. When we talk about muscular hypertrophy or growth, we're referring to increasing the number of sarcomeres in the muscle fibers of a given muscle, a process that can occur in one of two ways. The first method involves adding sarcomeres from one end to another and is called serial hypertrophy, whereas the second and predominant method involves adding sarcomeres side by side and is known as parallel hypertrophy. The former method affects the length of the muscle fibers, while the latter method affects the thickness of the muscle fibers. So, to wrap it all up, our goal when chasing muscle hypertrophy is to provide an external stimulus at the macro level, like lifting weights, that creates a cellular signal at the micro level that induces an upregulation in muscle protein synthesis so we can incorporate new myofilaments into new sarcomeres. Not too shabby, right?